welcome to this session, which is titled "Is Mitochondria the Missing Link Between Diabetes and Bone?" Today's webinar will be hosted by Paula, Guerra, who is working at the Molecular Endocrinology Department. She's an expert on the mitochondrion and how to assess mitochondrial function. And she'll be talking for 20-30 minutes, and then there'll be ample of time to discuss what she's saying. So please take it away, Paula.
So hello everyone, my name is Paula. As Morten mentioned, I'm a researcher here in Denmark at the Molecular Endocrinology Department, and I have been working my whole research career trying to understand mitochondrial function in different disease contexts. I will make the talk in two pieces, so we can have questions in the middle, and then we can discuss the different sections, and of course at the end everything together. If you have any questions, just write them in the chat and I will try to answer them as we go. Otherwise, we can have them live in the end of it. So, right. So my whole goal today is to convince you the mitochondria is the mighty mitochondria, and that is one of the most important things, or not the most important, of our cells and our bodies, not only us, all the eukaryotes has mitochondria, and they regulate a lot of functions. You all may know them as the powerhouse of the cell, but that's just one of the things they do. It's a very important one, but they also serve as signaling hubs, they integrate different signals, etc. So I will just go through a bit of basics of mitochondria, so then we are on the same place, because I imagine a lot of you have only studied back in the days in biochemistry, and everything is sort of a bit forgotten. So I will just take it from the beginning and just briefly remind you that mitochondria is an organ of the eukaryote cells that was incorporated from bacteria inside of one of the early prokaryote cells. And in that process, it was not only that the mitochondria will consume oxygen to produce ATP for the cell, it's actually a symbiotic process. So mitochondria had 5,000 genes when it was incorporated into the cell, and then it has lost cells that were, not cells, sorry, genes that were given to the nuclei to avoid double coding, but also it was incorporated proteins from the nucleus, and that was this symbiotic process that ended up being that the mitochondria only have very few genes left, actually only encodes for 13 proteins of the respiratory chain, and those are the ones you can see in this picture in dark orange or brown. And then of course they have their own replication machinery because they have to synthesize their own proteins. All in all, this process has still going on, and some people argue that eventually mitochondria will lose all their genes and have nothing left, but we'll see about that. So far today, the main role of mitochondria is energy generation, and that is done in a regulated way and in a controlled way. And many people think that that's controlled by the DNA in the nucleus, but that's actually not true, or at least in most of the cases, it's actually the mitochondrial DNA that regulates it. Mitochondrial DNA, as mitochondria was a bacteria, is a plasmid, it's a circular plasmid, encodes for their own replication machinery, but also encodes for these 13 genes of the respiratory chain. And you can see them in this figure on the right, in blue, NAD1, NAD2, and all the different ones. And those genes are replicated and also are transcribed and translated into proteins that they later are incorporated in the respiratory chain. Mitochondria doesn't have only one copy of mitochondrial DNA, as many bacteria have 100 to 1,000 copies, which means that changes in mitochondrial DNA are not easy to stay, because it requires that it has to be a change in a lot of different molecules in order for it to stay. That also means, as we'll see later on, that diseases caused by mutations in the mitochondrial DNA are not rare, are extremely rare. Also, if you think about a cell and one mitochondria or mitochondrion, that's not actually true. Mitochondria is a group of mitochondria that work as a network, so that creates another level of complexity. As you can see in the figure on the left, the main part of the mitochondria The main roles, as we know them today, is the energy metabolism. We get carbohydrate, fatty acids, that they get integrated and they produce ATP, we produce also CO2, and so on. Just briefly a bit more into mitochondrial DNA. Mitochondrial DNA encodes for 13 proteins of the respiratory chain, and you can see them in this figure on the right, that's marked with different colors. Those are the main ones of the respiratory chain, and as you can see, they are in almost all the complexes, except for complex 2, and they are very hydrophobic proteins. And that's one of the hypotheses where people argue that these genes were not lost to the nucleus is because they are so hydrophobic that being synthesized in the nucleus, in the cytoplasm, they have to be incorporated across the membranes. It will be a very complicated and inefficient process. That could be one of the reasons. Another hypothesis is that these 13 proteins control the efficiency and the amount of respiratory chain complexes by them controlling how much ATP is produced from the mitochondria. And that's what it comes about, that is a way for the mitochondria to regulate energy metabolism and as a master regulator or main switch controlling whether we go to produce ATP from the oxidative phosphorylation or respiratory chain, or we go through glycolysis or other pathways, which are less efficient, but they're also there. I know this is a bit basics, but I think it's always a good reminder how the respiratory chain works. That's the basics of the mitochondria. And for a lot of you, maybe this is so back in the years that it's always nice to see a little bit how it goes. The electrons goes donated from NADH. It goes through all the different complexes. The protons are being excreted from the matrix to the intermembrane spaces to the complexes. That creates membrane potential. This membrane potential is critical for every single mitochondrial function. So when you argue about difference in membrane potential, you argue about difference in signaling and function. We get ATP from complex 5 and we consume oxygen. And that's what will happen in normal conditions. However, if we have a mutation, let's say in complex 5, there's most key of them, and that doesn't work, what happens? The electron keeps passing, but they go a bit slower. The protons get also excreted to the intramembrane space, also a bit slower, because there's no ATP production, because complex 5 doesn't work, and the ATP production is either abolished or reduced significantly, meaning that there will also be a reduction in oxygen consumption from complex 4. And this will be the major or most critical situations. This situation can be that dramatic that there are very few patients or animal models with mutations in these complexes. This, we can link it to this symbol of the leaking pipe. So you can see the respiratory chain as a pipe that leaks. If you have a defect in the respiratory chain, whether it's from a mitochondrial DNA encoded protein or a nuclear DNA encoded protein, if something is not working, it doesn't matter how much water or energy you put through, it's going to leak, it's not going to be efficient, and this leak is going to eventually create problems. This leak could be related to reactive oxygen species. The respiratory chain has a byproduct that is reactive oxygen species with oxygen. Becomes water, it's not always an efficient process, and sometimes electrons are lost on the way, and we get mitochondrial superoxide or other reactive oxygen species. However, this situation is usually balanced and fine unless we have a defect. And when we have a defect, then this small leaks becomes bigger and bigger, and that creates a problem. Then we have oxidative damage, and that creates damage to all biomolecules, protein, lipids, or DNA, and eventually will lead to cell death. The most severe mitochondrial dysfunction we can think about is about chronic fatigue, and that's if we have this leaky pipe, then we're trying to get energy, we eat, we try to transform it in ATP, but then that doesn't happen. So then eventually we're having this severe chronic fatigue. When we talk about fatigue or exhaustion in these cases and in these patients, we're talking about situations where they can barely raise their arms, they can barely walk. Sometimes the walking distance they can do is maybe 500 meters or things like that. So we're talking about very severe discapacities. And this is another way to look at more molecular of what is happening when we have an impaired mitochondrial energy production. And that's the most common situation. We have a healthy mitochondria in orange in the left. All the complexes of the respiratory chain are in this top of the cristae between the in the inner membrane, connected the intermembrane space and the mitochondrial matrix. And they are in this cristae, so they're very close together, because electrons, they just fly very quickly. They go here and there, and they will just go wherever is easier for them to go. So then you can maybe think about, like, you have a cable, and then if you peel it, you will lose electricity everywhere. So in this case, what we do is we put them all together. So then the electron goes in the right way for us to get ATP and to consume oxygen and don't have so much reactive oxygen species. However, when something goes wrong, for different reasons, it can be a genetic disease or it can be external factors, these supracomplexes can be disassembled somehow. There can be more space. The cristae is not that packed together, meaning that the electrons will fly everywhere. They will get more ROS. Then there is less ATP. It becomes less efficient. We're having more leaks in our pipe. So it doesn't matter how much electrons we put through there, we don't get enough ATP. And then we have this imbalance between ATP production and ROS production. That's what we call a dysfunctional mitochondria in the general way. Mitochondria, as we said, is the major ROS production, and ROS is a signaling molecule, as we can see in this green square in the picture. That's redox signaling. Is very important. It will regulate absolutely everything, and it's one of the major regulatory ways. It also links mitochondria with another major situation, and that's calcium efflux. That's newly researched. We can talk about a bit later, where ROS efflux, from the mitochondria to the cell, and also calcium efflux becomes the major signaling pathways going together from the mitochondrial transition core. The situation with ROS is that if we have too much, because respiratory chain is not working properly, or we have any other issues, there's too much ROS, then it starts to, for example, mutate the DNA, and then we accumulate more mitochondrial mutated DNA. It can also lead to lipid peroxidation. Lipid peroxidation actually works as a cascade. So when a lipid becomes oxidized, it will oxidize other lipids, and also it can do it to proteins, making that maybe the respiratory chain that was inefficient already, it becomes even more inefficient, and then it creates more ROS. And then we are getting in this feedback, positive feedback. Mitochondrial dysfunction, that will be one of the problems, and when it gets so severe, we will end up having either apoptosis or necrosis, and both of them will lead to disease, but also it's a very common process in aging. Just to remind you briefly, mitochondria is a subcompartment from the cell, if we can talk about it, but it's not like other organelles where there's just one membrane. It's just a double membrane situation, so we have actually kind of three spaces. We have the cytosol, we have the inner membrane space, and we have the matrix space, and there's a whole crosstalk between them. Anything that goes wrong in this crosstalk, it leads to mitochondrial dysfunction, and it can lead to different diseases. Just briefly, if you look at the section A of the figure, you can see this cristae with all the supracomplexes and all of them packed together. The section B, you can see VDAC. VDAC is a major pore in the membrane of the mitochondria that serves to transfer a lot of different components. Then we can see that the respiratory chain in the figure C is also associated to the FAO metabolome. FAO is fatty acid oxidation. So respiratory chain is also connected to the oxidation of fat, another energy source. And finally, if we look at the picture D, we can see how the cristae is associated with the inner and the outer membrane, creating complexes. And these complexes can also get associated with the ER. So everything goes packed together. We tend to think about mitochondria as this single thing isolated here and there, but actually they're together because they're bacteria. They're groups of mitochondria, everything together, signaling to each other. And as you see in picture E, the way they create more mitochondria or we lose mitochondria is like bacteria through fission and fusion. So when we have, for example, damaged mitochondria, then we have a fission process that will remove this damaged mitochondria. Or if we have a cell cycle situation that the cell is going to divide, it needs more mitochondria, it will do the same. And then we have the process of fusion, where smaller mitochondria becomes one bigger one. This is all a whole dynamic process, and as I mentioned, it happens and generally, as you can see in the steps 1 and 2, because fusion and fission, that's happening all the time, this dynamic. And then, of course, if we have some damaged mitochondria, as this in step 4, it can be targeted and it will be degraded in mitophagy process. It can also be that this mitochondria gets recovered and it gets integrated again in the pool. All in all, these situations that is a different compartment, that it has its own DNA, and it has this dynamic process, makes it really complicated to understand what's going on and to relate it to different diseases. But also, this is one of the most cooler things that we can talk about mitochondria, is this whole dynamics. It's not something static, it's moving all the time. We have new mitochondria, they get incorporated, as you can see in the part A of the picture. We get damaged mitochondria, we get rid of it through mitophagy. But not only that, mitochondria also moves through the cell to talk with the ER, as you can see in the picture B, but also to the nucleus through reactive oxygen species, through NADH levels, AMP, because they produce most of the ATP. Also, AMP is part of their signaling. We have the UPR signaling, and all of that crosstalk between the mitochondria and the nucleus, then regulating everything. Then the nucleus will produce different regulatory proteins that will change mitochondrial function. And that's how this crossplay always goes. And that's why it's very difficult also to say that this is just happening on the nucleus, ignoring what's happening in the mitochondria, and vice versa. Here's just a picture when we talk about these dynamics. This is how a cell looks like. In this case, it's a dermal fibroblast from a healthy person. You can see in green is the mitochondria, and in blue the nucleus. You can see there are more mitochondria closer to the nucleus for this interplay we just talked about, and a bit less in the edges. But it's all these fibers going all the time, and this is just a picture. If we could see a video, we will see how they moved all the time. And that's what they make them great sensor organelles. Everything that's happening in our bodies gets integrated into the mitochondria. If we change our diet, if we do exercise, the mitochondria will react to that. Okay, we have more energy now or less energy now in forms of food. Let's process it faster or let's store it. We do exercise, we need more energy. Okay, let's produce more ATP. Also, it has related to different infections or chemicals, like the immune system is very much regulated through the mitochondria. If you have to react to an infection, that will be changing the mitochondria. And then it's also related to mental stress, because cognitive function depends a lot into mitochondria. Not only for the ATP, but also because, for example, glutamate, it gets produced in the mitochondria from glutamine, and thus becomes one of the major neurotransmitters. So everything is linked together in this beautiful connection of metabolic system, immunoinflammatory, neuroendocrine, all connected into the mitochondria. And that's why whenever you're studying any disease, you should look into your mitochondria and see what they're doing, and that will pinpoint of what's going on. This is just another way to see how things can be done or analyzed. For example, we have different circulating factors in the blood. We have lipids, glucose, we have also hormones. We have different inflammation markers, and all of those goes inside the cell and outside the cell, going through the mitochondria and changing gene expression. When we talk about mitochondrial function, we usually talk about how the mitochondria is working, what's going on inside. And when we talk about mitochondrial signaling, that's mostly inside the nucleus, because there have been changed some gene expression pathways. And another way to look at a mitochondria as an evolutionary situation, as a signaling organelles, we consider that there's different environmental changes that will alter mitochondrial function. For example, it will have a stream change, and then there will be a reduction of energy that could lead to disease or death. It could be that it has to produce more energy, and that leads to different high energy molecules, protein modification and signaling. Could also be that, okay, we are moving in this more homeostatic situation, we can adapt to it, and then we can activate different pathways in the nucleus. And the more adaptability the mitochondria has, the more it's associated with different healthy aging situations and longevity. And finally, I just wanted to show that the mitochondria works in different organs. I mean, all the cells in our body have mitochondria, but they are also very different. So the complications or the consequence of mitochondrial damage in each tissue is quite different. And that's what it makes it also very complicated to study in the whole organism, because it will not react the same way. For example, skeletal muscle is one of the most affected by mitochondrial dysfunction. When talking about weakness and fatigue, it's extreme weakness and fatigue, myopathy, and of course also some neuropathy. There, for example, the eye is another organ that is very much affected. So most of the patients that has any kind of mitochondrial disease will have some sort of retinopathy or optic problems. If you look at this drawing is actually from 95, so it's a bit old, we could say. And then there's no bone in there. And that's why I just glue it there on the right, because, okay, what's happening in the bone? Yeah, that's true. Most of the mitochondrial function has been studied in the liver, on skeletal muscle, some of it in the heart, but not so much has been done in bone. But of course, bone cells have also mitochondria. And here's where I would like just to make a small pause and see if you guys have any questions about this part, about the basic of mitochondria, before we move on to bone metabolism or diabetes, and we get everything a bit more complicated. If you have any questions, just unmute.
This conference will now be recorded.
As I just mentioned, when we have bone cells have also mitochondria that can be either functional mitochondria or dysfunctional mitochondria. And that will quickly make us think about, okay, if we have functional mitochondria, we have healthy bones and everything is fine, as we could see in the higher part of the figure. Or if we have dysfunctional mitochondria, maybe we have not so healthy bone, or maybe even an osteoporotic condition, as we can see in the lower part of the figure. So what can we find in the literature about this? So as you all know, bone remodeling is a very energetic consuming process, because it's happening all the time. There's always bone formation and bone resorption. So there's new cells being differentiated from the bone marrow into osteoblasts, and the osteoblasts into osteocytes to form new bone. Or on the other side, there is also monocytes that are being differentiated into osteoclasts that will going to reabsorb bone and remove it. And we also have macrophages over there and different structures. So this all in all consumes a lot of energy. It's a bit of a funny thought, because at least I tend to think about that bone is something static, it's just there and it's hard and that's it. It's actually pretty much alive. So as I just said, it consumes a lot of oxygen, and as we just said, it consumes a lot of energy. And I just said, fatigue is one of the major symptoms of patients with mitochondrial diseases, because mitochondria produce a lot of energy. So what happens when we have mitochondrial dysfunction and there is not enough energy? This balance between bone formation and bone resorption is going to be affected. A recent review has been published where putting all together this different signal, and in the part A of the figure you can see how mitochondria will receive strengthening signals that tell them, okay, this is the situation now, what should we do? Then we can have more ATP or do more oxphos, or we have other metabolites to signal what's happening. And depending on that, then the cells are going to be differentiated to osteoblasts or the osteoclasts. And it's in a general way, as I just mentioned, we have this balance between bone formation and bone resorption. When mitochondria is working fine, everything's okay, and we have this balance. However, when mitochondria is not working fine, we have an imbalance where there's less bone formation and more bone resorption, and that could lead to different diseases, and that's a common process in aging too. How is this mitochondrial dysfunction related, or what it is? Well, it's metabolic decline, for example. We can see it as oxidative stress, structural disruption of the mitochondria or the network, and quality control failure. The nice thing is that all those things can be regulated therapeutically. So that is a new interesting option to treat bone diseases. We can try to reduce oxidative stress, we can try to modulate different metabolites, all in all try to bring mitochondria back to this homeostasis where they can work and we can get our healthy bones. If we look at a bit more in detail, mitochondrial bioenergetics is going to regulate whether mesenchymal stem cells, the cells from the bone marrow, are getting to be differentiated into osteoblasts or not. There's a lot of different signalings from certain pathways, AMPK to PGC1 alpha, and all of them end up into the mitochondria to produce ROS or to produce ATP. If we produce more ATP, that means we also produce a bit more ROS, we are going to get osteoblast cells. It's not so clear why osteoblasts need a higher OX4 rate than other cells, but the reality is that mesenchymal cells or stem cells in general are very low energetic cells. They have a very low oxidative phosphorylation, but also very low glycolysis. And still, if we inhibit a bit more of that glycolysis and we increase the OX4, we will get osteoblasts. And that's why, again, mitochondria is key to all this process of bone formation and bone remodeling. We go back to the major topic, that's energy metabolism, and mitochondria is going to integrate the major source of metabolism, of energy metabolism. We have glucose, we have fatty acid, and we have glutamate. On the left side of the figure, in green, we can see glucose that gets processed through glucose 6-phosphate and to pyruvate. And then pyruvate has two options. Either it can go to the right and be integrated into the mitochondria and produce energy through the TCA cycle and the respiratory chain, or it can keep continuing the cytosol to be processed as lactate and be secreted. Both pathways produces energy, but there's a huge difference into the amount of energy they produce. The mitochondrial pathway is a lot more energy rich, will produce more ATP. When we have fatty acids will go into the mitochondria as acetyl-CoA fatty acids, will get processed through the TCA cycle, and then into the respiratory chain. That's another way of producing energy from fatty acid. The situation is that's a bit slow because it requires more steps, while pyruvate go a bit faster into the TCA cycle. And finally, on red on the right, we can see glutamine. Glutamine can be incorporated into the mitochondria, been changed to glutamate, and then incorporated into the TCA cycle again to produce energy to the respiratory chain. This is a very simplified version, but it more or less tells us that, okay, mitochondria, a lot of things have to happen before we reach respiratory chain, meaning it's a slower, more controlled option. When glucose goes to lactate, that's very fast, that's very quick, and because it produces very little ATP, when we have mitochondrial dysfunction and all our ATP comes from glucose, it's usually not enough. And that's why some patients with mitochondrial diseases will have lactoacidosis, because it produces too much lactate. However, in a normal situation, we can see a balance between one and another. Because as you see, fatty acids are incorporated or glutamine is incorporated into the mitochondria, or pyruvate, there are different signaling molecules that will be activated because we'll get different metabolites. So that's a way for the mitochondria to get, okay, this is what's happening now. We're getting energy, but we're getting it from here or here, and at the same time, we're signaling. For example, mitochondrial fitness is fine-tuning Wnt signaling. In these studies in human fibroblasts and in zebrafish models. Mitochondria is dysfunctionally or is reducing the ATP production because of different signalings. That will signal through calcium to the ER, and then the ER through the different signalings through CHOP and ER stress and everything will go and inhibit different signaling in the nucleus, and this signaling, for example, will be Wnt genes that will be reduced. And Wnt is a key pathway for bone formation. So that could be a way for the cell to regulate, okay, we don't have enough ATP now, we cannot do bone now because that consumes too much energy, and we have other things to do with this little ATP we have now. Another situation, as I mentioned before, mesenchymal stem cells are usually dormant, the mitochondria is not very active, very little energy consumption they do is through glycolysis. However, as long as you start the differentiation process, they get activated and then decide where to go. And in this case, for example, if we have more oxidative phosphorylation, we will have more osteoblast, and there will be bone formation. But what happens also when we have more oxidative phosphorylation? We have also more ROS. So that's why comes the part that the cell has to respond to the signaling and take care of the ROS levels. If we have a situation of pathogenesis where these ROS levels are not counteract, or we have a situation of aging, these ROS levels could activate adipogenesis or other situations. So everything is a balance. So we should not talk about, okay, this is good or this is bad. It depends where we are and what we're going to do with it. So it's all a balance. So now to go a bit more in details on this situation of what kind of mechanism is happening, whether this is happening only at cellular level or are we seeing this in bones, I'm going to go through different mouse models of mitochondrial dysfunction that we could use to evaluate the bone phenotype. So the first one of all is about complex 1. So complex 1 is the first complex of the respiratory chain. It's actually very big. It comprises a lot of different molecules. You can see here in the green, the ones that are encoded by the by the mitochondrial DNA. And the mitochondrial DNA is taking care of these very hydrophobic proteins. The complexes are not assembled as such. There are a lot of proteins who are just assembly factors. And you can see we start on the left with the endos 2 and 3 that will bind with 7 and 8, and they will go from the matrix into the membrane, the inner membrane, and then the inner membrane will have other proteins already there. And all in all, we're getting this whole assembly situation. So in this case, there's a mouse model where endos 4, which is not a catalytic subunit, is suspected to be an assembly factor, is knocked down, or knockout, sorry. So what happens to these mice? You can see in the left, when we have a global endos 4 deletion, the mice has alopecia and they're quite small. That's a characteristic also of most of these mouse models that you will see that they're smaller, and you can think about that they cannot produce enough ATP. So if they don't produce enough ATP, they don't grow. That's one of the simplest ways to see it. And if we look at the different figures in the part A of the figure, you can see how there is the bone formation is changed in the different tibia, both at the trabecular level and also the proximal tibia. And you can see in this case, it's okay. I would have expected that we would have less, but in this case, there's more bone volume. As you can see in figure B, and the trabecular number is higher in figure D. And if you look to C, D, E, and F, that's all the different bone parameters. Most of them are higher in these mice. If we look a bit of, that will make us think, okay, there's higher bone formation. That's a bit odd. We thought about, okay, we don't have enough ATP. Why we're having bone formation? That's a highly consumed process. Let's look at the osteoclast. And when we look at bone resorption, we could see in figure H, I, J, and K, how some of these, the osteoclast surface or the osteoclast number are reduced in this bone, in these mice. And if we look at figure L, there's also less size of osteoclast or surface of resorption in these bones. All in all, indicating that, okay, it's not that they're having more bone formation, it's that having less bone resorption. And why is that? Actually, bone resorption is a highly energetic process because osteoclasts consume a lot of energy to be active and to resorb bone. To resorb bone, they use a lot of ROS, and that means that they have to create a lot of ATP in the process and have the mitochondria fully working. And these mice cannot do that, and that's again an unbalance. So it's not that straightforward to say, okay, mitochondrial dysfunction, that means they're going to have less bone mineral density, for example. It's a whole balance of the process, and it can be affected at different points, depending on what part of the mitochondria is affected. In this case, it's complex 1. Actually, complex 1 is the major source of ROS in the mitochondria, meaning that it's affecting more osteoclast activity than maybe osteoblast. Another mouse model of a nuclear DNA encoded protein, that's the POLG. POLG is polymerase gamma, and is in charge of replicating and proofreading mitochondrial DNA. As we talk about, mitochondria has its own DNA, and it has their own replicating machinery. That could be, they're all encoded, funny enough, in the nuclear DNA. So when we have damaged POLG, then we have an accumulation of mitochondrial DNA mutations or a depletion of mitochondrial DNA, depending on whether the replication part of it is affected or is the proofreading part of it. Paul G mouse, as you can see in this figure, has been studied quite a lot, and they have studied this concept of aging quite a lot. So, for example, the mutation of mitochondrial DNA in the Turkish bar is the mutator mouse. It's called mutator mouse because it accumulates humongously amounts of mitochondrial DNA mutations, and then we can see that heterozygous is not that much. So the threshold between those two is considered the threshold of a healthy aging situation. If you look at the wild type mouse heart of brain, this is all in heart of brain, these three. In purple, you can see that it's very low. So Paul G in heterozygous condition, it adds more mutations, but not as much as it's homozygous. And then in blue on the right, you can see age human colon, and that's a way to study colon cancer, because then the colon is a very fast changing tissue, and that means there's a lot more mitochondrial DNA to replicate, and it accumulates mutation faster than other tissues like the brain, for example. This mutator mice, you can see it here. It was described in 2004 by Alexandra Trifonovic, and is the model of aging most studied as a transgenic mice. It's a mutation in the Paul G. It's a nuclear encoded protein. As you can see in the upper side is the mutated one. It's not only smaller, it also has a very severe kyphosis. And if you look on the right, there's a different levels of kyphosis. So not all of them are equally severe, but all in all, they all have kyphosis. And if the mice get a bit older, then we'll have a severe alopecia too. Just so we can look at the numbers, here you can see that the length in red is the mutator mice and in blue is the controls, that there's a shorter length, but then when we look at the lean mice correlated with length, it's not different. The percentage of fat is also smaller. That's funny enough, it's also a characteristic of a mitochondrial disease patient, that they have less fat, and they also have a lower bone mineral density. If we look at a recently published study with cells from these Paul G mutator mice, they can see that there's a decreased osteoblast densities and then a decreased mineralized matrix. As you can see in the A and B figure, it's a bit difference between female and male, but in both of them it decreased. In green, you can see the wild type, and in orange, the Paul G mutated mice. And you can see that it's not only decreases more, but also it's already started quite lower. So already at 4 months of age, what we could speculate that these mice have a peak bone mass, the peak bone mass is already a lot lower. And if we look at the figure C and E, we can see that the wild type in the upper side has a lot more alkaline grain staining. That's a measure of mineralization compared to the mutator mice in the lower part. And the same for ALP staining on the right, for the wild type on top and the mutant in the lower part. All in all, there's decreased bone formation in these mice. But there's also an increased osteoclast density and osteoclast resorption activity in these mice. So these mice are, in a way, really screwed because they have low bone formation and high bone resorption. So that's why they have this very severe kyphosis. You can see in the A, B, and C the different osteoclast ratio numbers and resorptions, and in D and E the quantification of the cells in images. And before moving to another animal model, the only thing we have to be a bit careful here is that we're talking about a global mutated mouse, meaning that also the muscle is affected, and the muscle is very severely affected. So that could be that the bone are not only losing activity, they're getting an unbalanced bone remodeling because the bone cells are affected, also because the muscle is also affected and the muscle is not pulling the bone and having the mechanical stress to activate bone formation. Another mouse model that we can see here is going to be now with energy metabolism. If we look a bit more detail in fatty acid metabolism, we can see that that's from the right in green. The fatty acid get incorporated from the extracellular space into the cytosol, and from there to the mitochondria, and we have these components of the fatty acid carnitine shuttle, and the CPT2 protein is mutated in these mice. And we can see here that there's male mice don't have any problem. It's only a small transient bone loss in figure C at 6 weeks of age. However, when we look at the females, here it comes, there's a more severe bone loss and it's maintained on time, but it's especially exacerbated at 12 weeks of age. It happens both at the tibia but also at the vertebra. But there's no changes in cortical bone, as you can see in figure E on the left. The in vitro differentiation of osteoblasts of these cells is also impaired. You can see the different ALP staining, and there's also thinking, okay, because female is affected, can we get it better with giving them estrogens? And that is not the situation. I think this is the last mouse model, if I'm not wrong. This is going to affect the glutamine, so that will be the red part of this figure. And glutamine is a very abundant amino acid, and it's not abundant because it's used for many things, not only, as you can see in these pictures, to go inside the mitochondria and produce energy, but also as a source of nucleotides and source of glutathione, etc. It's so necessary that it's considered an essential amino acid, even though we can synthesize it. There's this study from last year where glutamine supports the proliferation of mesenchymal stem cells and osteoblast differentiation. We can see in these mice where they have knock out the gene for glutamine processing from glutamine to glutamate, and we can see that there's a reduction of bone mass. And also, if we look a bit more in the cellular studies, we can see that there is an increased glutamine consumption and an increased metabolism during osteoblastogenesis. We can see it more clearly in the upper part of the figure. In the A and B, we can see the osteogenic media and the adipogenic media producing osteoblast adipocytes. And in E and F, we could see that what happens when we don't have glutamine, that adipocytes doesn't change that much, but osteoblasts, they are quite decreased. So all in all, these mouse models show that we have a situation with functional mitochondria and healthy bones, and dysfunctional mitochondria and not healthy bones. However, it's not clear that it's going to be in one way or another. It depends on where the dysfunction lies. We will have different bone phenotypes. So here we say, okay, this is what happens in mouse models. Is this the same situation? That's actually one of the questions in the chat, that patients with mitochondrial diseases display weaker bones. Well, let's see. When we talk about diabetes, we talk about also that there is a mitochondrial dysfunction. Also, there are some contradictory results. I will not go in detail into all of it, but just briefly show there are some studies with diabetic patients, both type 1 and 2, with diabetic nephropathy, that shows reduced mitochondrial DNA content, as you can see in the bottom of the table. Together with that reduction of mitochondrial DNA content, you can see that there is two different situations. Diabetic patients with no diabetic nephropathy on the left, on the bottom left of the picture, they have increased mitochondrial DNA, while diabetic nephrotic patients have a decreased mitochondrial DNA and a decrease of the consumption rate, which is mitochondria respiration, indicating that diabetic patients maybe not have a problem with mitochondria, maybe they are increasing it to cope with the disease, and then when they get so severe long-term, they have nephropathy, they will have mitochondrial dysfunction. And that could be one of the explanations why there's so contradictory studies with diabetes patient and mitochondrial dysfunction. Just to show a bit more, they took cells from the kidney to do a bit more studies into whether hyperglycemia was the cause, and they could see that at least in vitro, hyperglycemia has a toxic effect on renal cells and mitochondrial function. As we can see, mitochondria is integrated all these different signaling, especially within energy metabolism in this case. But we can also talk about secondary mitochondrial dysfunction, especially in diabetes, and that's something that's been described again in diabetic nephropathy, where we have a situation with high glucose levels that will activate different inflammation markers, as you can see in the upper part of the figure, MPL and complex C and everything. And then also glucose is also going inside the mitochondria, because we have higher glucose, we have higher pyruvate, and we overactivate mitochondria. Then we have higher ROS, and then we have cellular damage. So a way to avoid this is going through the glycolysis in the cytosol to lactate. So that could be a way that the patients cope with it, and then have this secondary mitochondrial dysfunction avoided by not using the mitochondria to produce energy. And that could be a way for treating these patients is towards mitochondrial medicine. It's very much going on in the last years and being very studied, and it's a way to have personalized medicine. It could be that without personalized medicine, it's a problem in diabetes we have now, that some patients go well, some patients don't, some patients have diabetic nephropathy, and it's not so clear why. However, if we find a bit maybe what's going on in the mitochondria, we can find out what's to treat them better. And this could be also a way to have a response to different stress factors. Our mitochondria can be very good and have this normal response, or if you have to run a bit, you can run. But then you have also this age mitochondria, for example, that cannot adapt to different stress levels. But it can be treated in different ways to have a better mitochondria and then a healthier situation. And that's again goes with this symbol of the leaky pipe, that you have to see what to do, because that's not for putting more water into a leaky pipe, you will have a better result. You have to fix this pipe somehow. So those mitochondrial dysfunction contributes to the bone phenotype. That's where we go to talk a bit of patients. And in this case, we're talking about a rare disease patient, because mitochondrial diseases, as primary mitochondrial diseases, are very rare. It's one of the less abundant diseases. And then just a reminder of primary and secondary mitochondrial dysfunction. Secondary could be the case of diabetes. They have something else causing the disease, and the disease is causing mitochondrial dysfunction. And those are the ones you can see in blue in this picture. And then you have more primary causes, and the ones you see in orange, where mitochondrial dysfunction is causing the disease, like mitochondrial diseases. The clinical features of mitochondrial diseases are very mixed and complicated, and that's because we have different amount of mitochondria, mitochondrial DNA, etc. Overall, there is neurological and non-neurological ones. The neurological ones are the most severe ones. They have peripheral neuropathy, myopathy is very severe, cognitive function is very depressed. But if we look at the non-neurological ones, there at the bottom we have short stature, bone marrow failure, kyphoscoliosis. Okay, something going on with the bone. This severe changes in phenotype is mostly due to heteroplasmia. And that means that it depends how much mutated mitochondria or dysfunctional mitochondria we have compared to normal. We'll have different situations, and especially in the case of genetic diseases that has mutations in the mitochondrial DNA. We have patients who have almost no mutations and in the upper part, then they go fine, they don't pass a threshold. Then they have this intermediate where they have starting to have a bit of mutations, so then they will have some symptoms, and then the very severe ones with a very heavy load of mitochondrial DNA mutated, they will have the most severe symptoms. There's a study done in the US, in Philadelphia, where they studied the prevalence of risk factors for poor bone health in mitochondrial disease patients, and they could find that there is a high prevalence. So if you look at the upper part of the table, patients with at least one risk factor are 73% of the total, with two risk factors, 63, and with at least three risk factors, that's 41. So that seems to be that these patients are at higher risk of poor bone health. And this could be correlated to this heteroplasmic situation, that the more severe they are, the more factors they accumulate. In this case, we're going to focus on one mutation, just to give us an example of the mitochondrial DNA 3243A2G that affects the leucine tRNAs of the mitochondria. There's patients who are completely asymptomatic if they have a low load. There are patients with mitochondrial inherited diabetes and deafness. That's a sort of medium phenotype. And the last one has mitochondrial encephalomyopathy, lactic acidosis, stroke-like episode, or MELAS. MELAS is a very severe neurological disorder, and if the symptoms show up early in life, they will not reach the 40s. That's also why it's a bit difficult maybe to study bone in these patients, but let's see what we can find. When we talk about carriers of N3243 A2G with insulin resistant in not diabetic carriers. So as you can see in this figure, the blood levels during an GGT in these patients versus the matched controls, the patients are in red, and you can see the glucose levels higher, the insulin, C-peptide, and lactate. Everything is going on, even though they are not diabetic. If we look at the DEXA scan or HRP-QCT of these patients, we can also see that there's a huge difference between patients and controls, indicated that the, for example, total bone density, it's lower in these patients than in the controls. Finally, just a brief thing, we have a patient with a Lake syndrome, which is a mutation in the mitochondrial DNA. And they have been treated with benzafibrate, which is a booster of mitochondrial function. And in this case, there's a positive effect on osteogenesis for these patients. So that could be a future treatment for the patients with mitochondrial diseases to improve the function. As a final take-home message, there is a connection between diabetes, bone phenotype, and mitochondria. All in all, it's not so clear what is the mechanism, but something is happening here. So whenever you're studying bone or diabetes, take a look at the mitochondria, see what's happening, and maybe it will shed some light in your research and help us, these patients. Finally, just a reminder, that's the mighty mitochondria. And I hope I have convinced all of you that the mitochondria is the chief executive organ of the cell and is the one who's truly ruling in here.